Hello and good morning, students. This is a recorded lesson intended for Saturday, but because on Saturday you'll be having POE, so I'm not going to disturb you doing a revision on Saturday. So this is a recorded lesson which you will view and watch on Sunday. Okay, so that Monday we can start fresh. Okay, so we have done number 1 and number 2. Now we will look at objective number 3. Describe the role of gibberellins in germination of barley. And on top of that, you are required to have a look at 16.3 and 0.4. Yeah, objective 16.3 and 0.4. So the keyword here is only using describe the role of gibberellins, and it's only in germination of barley. Okay, now if you look at the previous learning objectives, okay, this one here, as you can see, gibberellin has many functions. So the same thing with auxin, we have done auxin already. Gibberellin also has many functions. So as you can see, for your syllabus, we are required to learn seed development and germination. This is for the syllabus. But then you have to understand as well that there are also other roles of gibberellins, such as stem elongation, just like auxin. When you look at auxin here, okay, also stimulate stem elongation. And other functions as well, like pollen development, it says here, pollen tube growth, fruit growth, and so on. Okay, so just to let you know that there are many functions of this plant growth hormones: auxin, cytokinin, gibberellins, and abscisic acid. Cytokinin is not in the syllabus, but even abscisic acid is also involved in closure of stomata. Okay, as you can see here, stomatal closure. Okay, and that is already done for abscisic acid. You must know the mechanism as well. Right, so let's look at, let's go back to the learning objectives. Okay, so what is this objective 16.3.4? So describe the role of gibberellins in germination of seeds. Okay, and then what is this role in 16.3.4, which we're just gonna talk very, very briefly, because we will talk about it in great detail when we reach this topic number 16, okay, which is on inherited change. So what is the 16.3.4? Okay, so describe how gibberellin activate genes by causing the breakdown of DELA protein repressors, which normally inhibit factors that promote transcription. So this, we're just gonna talk about this very, very briefly later on. It's only one slide, and we will learn in great detail when we reach this particular topic, topic number 16, objective 16.3 and 0.4. Okay, so now let's look at gibberellins. It's going to be very, very short. So the learning objective is this one again. Describe the role of gibberellins. So don't forget, the keyword is describe. Okay, the keyword is being used here, describe. And then it's only germination. Okay, germination. So germination means seeds, how seeds grow, how seeds begin to grow. Okay, so gibberellin or gibberellic acid, okay, it's also known as gibberellic acid. Some background about this: they are found in plants. They are also found in algae, fungi, and also some bacteria. Okay, they've been found in plants. They also can be found in fungi, algae, and some bacteria. Okay. In flowering plants, it is thought that gibberellins are synthesized in young tissues of the shoots, in developing seeds, and also small amount in leaves. Okay, and this is the chemical formula. Okay, just some names like gibberellic acid, for auxin, IAA. Just want to Sometimes you tend to forget these names. I mean, the short form for auxin is IAA. Don't forget that, IAA. Okay, IAA, short form. Gibberellins, GA. That's the short form. Okay, now the role of gibberellins in the germination of seeds. So we have to learn the internal structure of seeds, before we talk about gibberellins in great detail. So, but let's read this one first. The role of gibberellins in the initiation of germination of cereal grains has been studied detail. Seeds. So seeds contain an embryo and stored food reserved. Here, the food reserves are stored in a mast tissue called endosperm. The main reserves are starch. Okay, so the main reserve is starch. It actually depends on the species of seeds. When we talk about barley or grains, it is starch. Sometimes, normally grains has the three macromolecule. It contains just a different color here. Okay, starch, which occupies the most central region of the sperm. Proteins, meaning the outer layer of the sperm, aeron layer. Before any seeds can start to grow, its food reserve must be hydrolyzed. So before any seed can start to grow, its food reserve must be hydrolyzed into soluble forms such as glucose and amino acid, so that they can be transported to the growing parts in the embryo. So actually, seeds will normally seeds contain carbohydrates, which is starch, proteins, and as well as fats, lipids. So normally seeds contain these three: starch, protein, and lipids. But when we talk about grains, it has more starch. Starch is the main food reserved. Protein as well is present, and actually fats are also present. Okay, that is in general. But when we talk about grains, starch is the main food reserved, and proteins, which are found in the aeron layer. Okay, aeron layer of the endosperm. So let's study the internal structure of this grain or seed, and this is using barley. Okay, barley. So barley is a type of cereal grain. Cereal, bijirin. In Malay we call it bijirin. Things like padi. Okay, these are all cereal grains. Wheat, barley, oats. There's another one which is wheat, which is used to make bread. Bread. These are all known as cereal grains. Okay, cereal grains. So mostly when we look at these cereal grains, we look at the structure here. Now this is the embryo. The embryo. So the embryo is the most important part. The embryo is the one that will grow into an individual plant. Okay, this is the one that will grow into an individual plant, and this embryo is the one that is gonna receive the nutrients from the endosperm. Okay, from the endosperm as well as from the aeron layer, which is proteins. Right. So let's learn the parts. This is the endosperm. This is where starch is present. So don't forget, it's not only starch, but it can also contain lipids, okay, as well as proteins. But then the aeron layer also has protein reserves. Outer layer of endosperm, protein reserves. Okay. Aeron layer. So aeron layer is actually part of the endosperm. So that's why when we say endosperm, in general, when we say endosperm, we say that it contains fats, protein, and starch. But to be more specific, the protein is present in the aeron layer, because the aeron layer is part of the endosperm. It's the outer layer of the endosperm. And then the seed coat. Okay, this is the seed coat. Don't forget the seed coat. Covering, which provide protection, yeah. Okay, protective, says here, protective layer. You don't need to know this coleoptile, cotyledon, young root, no need, yeah. So just need to know the embryo. This part is the embryo. Okay, now this is the sequence, as you can see here. In order for starch, in order for seeds to start, in order for seeds to begin the process of germination, it must absorb water. Yeah, water. Water must be absorbed. So without water, germination cannot start. Okay, so first, the uptake of water by the seeds, absorbed by the embryo, by the seeds in general, will activate the the synthesis of gibberellins. And gibberellin is synthesis, make sure that you know this, is embryo synthesizes gibberellin. That means the embryo is the one that makes gibberellins. And then the gibberellins will diffuse to the aeron layer. Aeron layer. Now, why aeron layer? Because the aeron layer has proteins. And examples of proteins are, one of the main examples of proteins are enzymes. Yeah. So the gibberellin layer will diffuse, the gibberellin will diffuse to the aeron layer and will make enzymes. One of the important enzymes, of course, amylase. Okay. Other important enzymes include maltase, lipase, okay, and proteases. Yeah, some enzymes that break down protein, proteases. Okay. So here, what is only shown is amylase, because because we want to show the breakdown of starch. So gibberellin, so the seeds absorb water, the embryo makes gibberellin, gibberellin diffuse to the aeron layer. Why in the aeron layer? Because we said just now the aeron layer has is where proteins are stored, and examples of proteins are enzymes. And so from here the enzymes can be made. Yeah, enzymes, amylase. So the starch can now be broken down to maltose by amylase, but don't forget the maltose is also, because this is still disaccharide, yeah, disaccharide is still quite big. So the maltose will be broken down to glucose plus glucose by maltase, and this maltase is also this maltase is also coming from the aeron layer, because maltase is an enzyme. Okay, so then the glucose. Now what happens to this glucose here? This glucose now can be absorbed by the embryo. Yeah, absorbed by the embryo for what? For cellular respiration. Okay, for cellular respiration. So it can start to respire, and then it will also begin to absorb some proteins, and which is important for growth. Okay, so germination is the growth of an embryo to young plant using the food reserve present in the seed. When the plant starts to produce its own food, that means when the seed begins to make leaves, okay, meaning the seeds grow and then starts to make leaves, then it will be able to make its own food. Yeah, then it will not rely on the endosperm anymore. Yeah, so when the seeds begin to produce leaves, grow into a plant, have shoot, then stem, and then have leaves, then it will not it will not get the food from the endosperm anymore, because it can now make its own food by photosynthesis. So germination will be over. All right, the sequence is like this. Yeah. So seeds absorb water. Gibberellins are produced by the embryo. Very, very important. By the embryo. Diffuse. So we use the word diffuse across the endosperm to the aeron layer. Gibberellins switch on the genes in the cells of the aeron layer, which cause them to use some of the protein reserve to secrete several enzymes, including amylase. You can see here several enzymes. Okay, so what is shown here is only amylase. Okay, it's shown here is only amylase. Don't forget, it also can make maltase, proteases, lipase. Yeah. In particular, amylase converts starch into maltose, and then maltase, see here again, maltase converts the maltose into glucose. The glucose will diffuse into the embryo and used for respiration and for the formation of macromolecules such as cellulose and so forth. Okay, so basically this one here is very, very important here. See, this is already very nicely done, the sequence. Okay, absorb water. Gibberellin is made by the embryo, and then the gibberellin diffused to the aeron layer to produce alpha amylase. And don't forget other enzymes. Okay, not only alpha amylase, they also make maltase, they also make lipase and other proteases. Okay, and then starch reserved in the endosperm hydrolyzed to soluble sugar. Soluble sugar means monosaccharide, which is glucose. Yeah, glucose. So starch into maltose, maltose into glucose. But you have to name the enzyme. Starch by amylase into maltose, maltose into glucose by maltase. Okay, and then it will be absorbed by the embryo, and that's it. Okay, this is also again taken from another textbook. As you can see here, nicely written down here. Starch into maltose, maltose into glucose. Okay, this is taken from a university textbook. Okay, so basically it's the same, yeah, just to double check. Okay, so in I think in O level, if you I'm not sure, because when I taught O level before, the radical will become the roots, yeah, and then the pumal, there should be pumal here, this is the pumal, this one will become the shoots. Okay, will become the shoots. Okay, radical will become the roots, the pumal will become the shoots. Okay, right. Right, this is the one that the learning objective 16, yeah, 16.3.4, yeah. So we're just gonna look at this very, very briefly. It says here, gibberellin cause the effect by regulating genes involved in the synthesis of amylase. In barley seed, it has been shown that application of gibberellin causes an increase in transcription, messenger RNA, amylase. In this, it has this action by promoting destruction of the other proteins. Okay, what does this mean? Doesn't make sense. Okay, let me show to you just just an overview of what it means by this. Yeah, so naturally, what happens is that when there is no water, there is no transcription of the gene that makes the amylase protein. This amylase is a protein. It comes from messenger RNA, which comes from a gene. Yeah? So this is like the gene for amylase. Transcription produces messenger RNA, and then the translation of the messenger RNA for the amylase will make the amylase. The gene for amylase, transcription, produce messenger RNA, and then translation of the messenger RNA will make the amylase protein. Okay. So when there is no water, there is no gibberellin, isn't it? Yeah, no water means no gibberellin. So at that moment, this pathway is inhibited. This pathway is inhibited by these DELA proteins. So DELA protein inhibit the transcription of the gene which produces the amylase. Okay, so that is in a nutshell. All right. So when it absorbs water, okay, water is absorbed by seed. This will synthesize gibberellins. Okay, gibberellins. Now gibberellins will destroy this DELA proteins or will inhibit this DELA protein. So water absorbed by seed, the gibberellins will inhibit or will destroy this DELA protein, okay, and therefore this pathway can start, and that's why the gibberellin diffused to the aron layer will switch on the gene for amylase, the gene for lipase, the gene for maltase, yeah, and many other enzymes undergo transcription to produce messenger RNA, and then the messenger RNA will undergo translation and it will make amylase and other enzymes. Okay, so this is the meaning of that this objective 16.3.4. Now we will look at this in more detail when we talk about topic number 16. Okay, so this is just in a nutshell. Okay, this is what it means by this short paragraph. Okay, so I think that's it for the germination. Yeah, the germination of seeds. When you receive this slide, you can read the rest. Okay, as you can see, just to summarize what we have covered so far, we talk about auxin, we talk about gibberellins. For auxin, we talk about stem elongation. For gibberellins, we'll talk about there is no mentioning of seed here. Okay, gibberellin for your syllabus, it should be germination, seed germination, but it is not shown here. So auxin, just to stimulate stem elongation. Remember, this is the one that is in your syllabus, stem elongation. For gibberellins, it is this one here. Stimulate germination in seeds by stimulating enzyme production. So that is the one which is in your syllabus. And don't forget, abscisic acid is the closure of stomata. That is one that is also in your syllabus. Okay, the rest is not mentioned in the syllabus. So I think you can read the rest. I think that's about it. For this particular learning objective, I did say that earlier in the lesson that this will be a very short learning objective. So basically, we have done topic number 16. Topic number 15. It's done. So the next one on Monday, we will slowly begin to talk about topic number 16. So 15 already done, and then 16 will be our next topic. Yeah, on inherited change. All right. That's all for today.